Okay. Mm. Thank you very much once again. This is Teacher Michael going through with you biology, senior 4, my dear students. We were looking at patterns of inheritance and we were supposed to define the terms that we shall meet in this subtopic. And the 8 terms I left you with was first filial generation, which first filial generation, which we write it as F1 generation. The F1 generation, first filial generation. So what is it? What did you find? Because it was an assignment, you should be having the answer with you, right? The answer should be with you. I'm just supposed to, or I leave it because you have found out the answer. First filial generation, you are saying these are offspring produced by crossing breeding. Let's say the best word, breeding, offspring produced by breeding, two homologous parents with the contrasting characteristics. Two homologous parents with contrasting characteristics. This term, it will be a little bit harder for you at the moment to grasp, but I would like you to pay attention. What do you mean by homologous parents? This is a term which I want you, and the contrasting characteristics. These are the key terms. I would like you to pay attention. I will come back and I explain more on this term. So term number 9 is the second filial generation. Or we call it F2 generation. These are also offsprings. But which offsprings are they? These are offspring produced by crossing two members of F1 generation by breeding. These are offsprings. These are offspring produced by breeding. By breeding two members of the F1 generation. F1 generation. I told you, pay attention. Next, I will emphasize on this when in the next part of our lesson here. Let us look at number 10. Number 10 is homologous allele. Sorry, not homologous, homozygous allele. Homozygous alleles. What are homozygous alleles? I said this is a pair of identical allele at a given locus. identical allele. At a given locus. Locus, when we talk of locus, we mean the position where a gene is found, a particular gene is found on a chromosome. We said a chromosome is found in the nucleus of a cell. You remember when we defined a chromosome in cell division. We said a chromosome is found inside the cell, and a chromosome is a long strand of DNA, that is deoxyribonucleic acid. It is an acid, as I explained the other time, an acid like those ones which taste sour. But this one is a complex one that determines characteristics, and they are only found in the nucleus, not anywhere else. So locus, so only this, let's look at it. So if we are saying, we can look at a pair of an allele. So if you have, we are saying, if this part, this drawing here, this illustration we are looking at is as an allele. An allele must have an alternative form of a gene. You can have an allele X, I mean another allele also, to take it to be Y, Y now. These two are allele. They form a gene. They form a gene. So if at this point we have A, and at this point also we have a gene A, so you'll find that X and Y are homologous. X and Y are homologous. In terms of the gene A, the gene which is, I said genes are represented by letters. Either it can be capital letter or small letter. Then we can have another, this is lowercase letter A, lowercase letter A. So all these positions, we can give them these positions. These are the different locus, different loci of genes, where genes are found. Genes are very many on a chromosome. So where a gene is located, that position is what is called a locus. So on a chromosome, we have several loci. So we are saying a homologous alleles is a pair of identical allele at a given locus. An allele here is A. The allele A and A on X and Y. The other are identical. So they are A and A is homologous, and A is also homologous. So you find that A and A, or A and A are homologous allele, are homologous allele. They are homologous allele. P and Q we can give us. So that is number 10. We can have another one, heterozygous allele, which is the opposite, heterozygous allele. Heterozygous allele. Now, heterozygous allele, we can simply define it as a pair of contrasting allele at a given locus. A pair of contrasting allele at a given locus. Let's take, for example, alleles. We are saying alleles are genes. Don't forget, alleles are genes, and genes are located on chromosomes. So if X and Y are chromosomes, these X and Y chromosomes, then PQ are also chromosomes, but they have a pair of identical alleles. So these alleles are homozygous rather. They are homozygous. Then we have heterozygous. Look at them, heterozygous. If I have another gene, K, not a gene but a chromosome K, then K, this is a K, then L, for example. They also have loci on which genes are found. If this is capital A, then this is capital A. This now, they are not the same. A pair of contrasting. If I have another gene, let's say I have another gene, R and then A. These are not contrasting. Not contrasting at all. Not contrasting, and therefore can never be homozygous or heterozygous, whatever the case may be. Can never. All of them not contrasting. Please forget about such things. So a pair of contrasting allele at a given locus. You will understand this thing more when we proceed. So those are the terms I told you to look at. So let us look at now a certain guy, a man who lived like more than 200 years ago. More than 200 years ago, and he was an Austrian monk by the name Mendel. Mendel and the laws of heredity. 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 So briefly, Mendel was a monk. A monk is like a priest. A priest, a Catholic priest is a monk. A nun is a reverend sister. That is a nun. But this is a monk, a male one. A priest can never be a female in the Catholic faith. So Mendel was a monk. Is a guy who struggled very hard to be a scientist, even to be a teacher. Very bright. He's a genius. He was a genius, whose work we are still looking at up to now, and is the father of genetics, if we are to look at. So briefly about that, he failed several examinations to become a teacher, like me. But this failing, I think, was not genuine, though. So, but he resorted to doing research with the statistical data. For the first time, nobody told him. No one taught him. So was he not a genius? So if I would like to take example of Mendel. You can be looked at as a very lousy person all the time, lousy, but you are gifted somewhere. People may fail to understand your worth, but proceed. Time will come when you will really be looked at, and you will succeed, and you will be powerful. Right now, we are considering him as the father of genetics, yet they looked at him as a failure during his time when he was struggling to become a teacher. Now, you can see his work here. So, be like Mendel. What did he do then? for us to praise him to do what, and give him as the title, the Father. Mendel carried out several experiments, and the experiments, some of the experiments he did were on a tree, a plant called Pisum sativum. Pisum sativum. This is what we refer to as kao. Mark my word. When you are told to go and buy cowpeas in the market, that green piece there you see is the seed of what is referred to as the Pisum sativum. And that is what Mendel carried out his experiments on. One of the experiments he did was on the height of this height of Pisum sativum. Pisum sativum, the height of Pisum sativum. In this plant, there is two characteristics, very prominent characteristics. One characteristic of the height is that they are plants which are tall. Then we have plants which are dwarf. Plants which are tall and plants which are dwarf. And these plants which are tall, if he breed tall plant with a tall plant, breeding tall plant with tall plant, that is dashing pollen grains on one plant, this one plant which is tall here, and then another one is tall here, you cross-pollinate them. Then the results come tall. Then those ones which you planted again, you cross-pollinate, you become tall. So what he did was, this one here was pure breeding tall. You continue breeding, a tall one will give you a tall, all plants tall. Dwarf one also, pure breeding dwarf. Pure breeding dwarf plants. Pure breeding tall plants. This is what he did. He said, fine, after realizing that, after cross-pollinating the same characteristics, tall, cross-pollinating after several generations, he called them, these are pure breeding tall plants. Then they are also pure breeding dwarf plants. When he did that, he said, fine, let me now do something. If I cross-pollinate the tall plant, pure breeding tall, with the pure breeding dwarf. All this, remember, we're talking about pure breeding. Pure breeding. This one is also pure breeding. After cross-pollinating this, the result Mr. Mendel got were all tall plants. Tall Pisum sativum. Tall, all of them were tall. Then he said, my God, what an interesting results. All the plants were tall. What said, what happened to this dwarf one? A question came into his mind. What happened to these dwarf plants? He asked himself. As a scientist and a genius, he looked for an answer. Said, fine, this thing I'm going to do. Then he self-pollinated these plants, the products he has got. So he said, okay, if tall is self-pollinated with another tall, with this tall one. But for the second one, this is the second generation, tall plants. These siblings, these offsprings, after self-pollinating them, what he got was interesting. He got a mixture of plants. Both tall and dwarf. both tall and dwarf plants. Tall and dwarf plants. In the ratio of 3 tall to 1 dwarf. According to him, he got them, and imagine he counted all of them, and then he got the ratio of the plants he has got. This gave him, I said, oh, it was interesting for him. He noted down this. But 34 years after his death, that is when some scientists came, a geneticist came across his work. By mistake, he read through and he said, this work was very interesting. That is the thing I'm now teaching you. 34 years after his death, at the beginning of the last century, he got these things, and then he wrote for us. Now we are reading, and I was also taught, I'm now teaching you as well, because the work of the father of genetics, who cannot teach you, and how can you not learn? Then, after that, using that, he came up with his own law. From this ratio, he came up with Mendel's first law. Mendel's first law, which states that Mendel's first law, from the ratio, he came up with the law. Mendel's first law, which states that the characteristics of organisms are controlled by factors. By factors which occur in a pair, of which only one is represented in the gamete. Only one is represented in a gamete. Any gamete, only one pair. Only of the pair, only one is represented in a gamete. That was his first law, and that is the law we are going to be looking at. And that is the law that we shall base on to do our genetic crossing or breeding, whatever the case may be. So let us look at presentation by use of symbols. This work, Mendel's work, presented by use of symbols. Now, what happened what do you think will happen here? What happened here? Why didn't we have, if we have a tall plant, tall, and then a dwarf plant, Why didn't we get the intermediate? Why didn't we get the intermediate here? It was not God. I put a cross here. It was not God. We got another toll. All the plans were told, like the one of the parents here, like the toll plan. All were told. It was this one which was God. There was no intermediate plan. What could be the reason? No intermediate was God. This intermediate was not God. Intermediate. Was not God. There was no intermediate produced. It was toll plan, like here, toll plan. So the question is, what is the reason for that? What is the reason why there was no intermediate plan? All were told in the first generation. All were told. Why? Told because, so the toll plan was because this dwarf, the gene controlling the dwarf, the allele controlling the dwarf condition, is recessive. Remember the term recessive. The gene controlling the dwarf condition of the pisum sativum is recessive. This one is dominant. Then this one is a recessive gene, recessive. One, it is combining with the tall one. The dwarf condition will never be attained. Will never. It will be suppressed. It will be tall. It will be suppressed. So the tall condition is dominant, suppressing the dwarf condition. So in the presence of the tall, dwarf will not appear. It will not express itself. This one is tall, then this one is dwarf, then they are. Let us mix so as you get the intermediate between tall and dwarf. No, tall. Don't disturb. You cannot do anything when I'm present. You can't do nothing when I'm present. I hope you get that, what I'm meaning. So this dwarf condition, as we have seen, because it is silent in the F1 generation, this we call it the F1 generation, F1. Why? Because we have said an F1 generation is the offspring of a breeding between two contrasting allele. Two contrasting allele. There is allele for this. So here, now, the mixture told what we have said we got a mixture of plants. So let us now see how we can present these things, the dwarf condition, looking at the tall and the dwarf. So we are going to use symbol, as we have said, using symbol. I told you in genetics, genes are represented by symbols. So allele for let the allele for dwarf condition. Dwarf condition. B small t. B small t. Then that for tall condition. B capital T. Why have we chosen small t and capital T? We have already told you that this gene here is a recessive. A recessive gene is represented by lowercase letter. And a dominant allele is represented by a capital letter. I'm talking of genes because an allele is a gene. An allele is a gene, an alternative form of a gene. So I'm right to say so. The allele for dwarf condition is small t because it is recessive. Then that for tall condition is capital because it is dominant. Now, if we have gotten that, let us now look at how we represent these things. We said at the beginning, the first thing now we are going to look at is the parent. The parent we got there, the parental phenotype. Parental phenotype. Parental phenotype. Phenotype. What is a phenotype? What did we say? What was the definition of a phenotype? If you were attentive, hey, you go back, find out what a phenotype is. The phenotype, we said, one is tall, and this cross here, it shows that this is breeding, this cross here. Then the other one is dwarf. Dwarf. You get that? Dwarf, tall and dwarf. These are the phenotype, observable characteristics you can see. But what brings about these characteristics which you can observe is the thing which is inside, and it is controlling the thing we call factors. In this case now, the modern term, these factors are now the genes or the alleles themselves. So this parental phenotype, then we have the parental genotype. Genotype. We have said characteristics of organisms are controlled by factors, which we said these factors are genes. And if the genes are the alternative form, they are alleles. So the alleles controlling this, they must operate in pair. Already told, we said the allele is supposed to be a capital T, but they are supposed to occur in pair. So we are going to have T and capital T. You cross now, there is no crossing. We have done it the first time there. Then, how about the parental genotype of this dwarf plant? It is a small t, small t. Why? We are doing this because the moment it is a capital T, small t, that one will be a tall plant. So this one here, we call it, these ones are homozygous, a pair of identical allele. Heterozygous, we shall look at them here. This is heterozygous, homozygous, tall, homozygous, tall. Look at the definition of the homozygous and heterozygous. Then, next, we are going to look at now meiosis. When meiosis occurs, we said meiosis leads to significance of meiosis. You remember, leads to formation of genes. Formation of gametes. Formation of gametes. So these are gametes now. We are going to form gametes. Gametes are separated now. Then we have gametes here, and then also we have these are gametes, then gametes. But these are now gametes. But the process that leads to formation of gametes, we have already looked at earlier on in cell division as being meiosis. Meiosis leads to formation of gametes. So this process, this arrow here, is meiosis, but leads to formation of gametes. When gametes are formed and breeding happens to occur, there will be fertilization. So this there we can have now fertilization. This will bond with this. We shall have, when it combines with this, write it with capital T. And then this one also with the capital T, small t. And then we have this capital T, small t. Capital T also and small t. Very good. These are, what are these we have got? These are the genotype. Just like they are occurring in pair. They are occurring in pair. These ones are occurring in pair. So this pair, meaning are they still parental genotypes? No. after fertilization, you do not form parents again. What do you form? You form the offspring. Now the offspring are the ones we first call the F1 generation, because these were contrasting. So this will be called F1 generation. F1 generation genotype. F1 generation genotype. So I would like you to tell me now the phenotype of these organisms, these offsprings. Phenotype. Phenotype. I see they are all the same. Capital T, small t. Capital T, small t. Here, I would like you to tell me whether these plants, the offspring of these two, whether they are tall plants or dwarf plants. The answer is here. We said all the plants, all tall, F1 were all tall. They are there. We have written it there. F1, all tall. Tall plant, all of them were tall plants. Tall plants, all. So the tall plant, now, so meaning all are tall. All are tall. Tall plants. Very good, all of them are tall plants. Now, being tall, why are they saying they are tall? Yet there is this small t here. Why are they intermediate? Because when you mix something, there should be something coming in the middle. Why? You have the answer with you. Don't you have the answer? I have told you. I said this tall condition, the allele controlling the tall condition is dominant over allele controlling the dwarf condition. So when the two are present in a gamete, when they are present to determine the genotype of the organisms, that organisms will outwardly appear tall, because tall is dominant. It will suppress the dwarf condition gene. So that's why these are tall, but not exactly the same with this. You see the genotypes now vary. Tall but with a very big but at the capital letter. So next, let us look at the next one. The next one. The next one. The next. We are going to self the plants here, but selfing the F1 generation. Selfing the F1 generation. Selfing the F1 generation. We have the parental genotype, phenotype first. Phenotype. We have here, if we look at the use of symbol, if we have put this means male, then cross it with the female, which is, these are the genetic symbol. This is female. So the parental genotype, phenotype here, we are saying this is tall, and then this is also tall. Parental genotype. Parental genotype, we have here T, small t, capital T, small t. Then we have parental the gametes now. Gametes. The gametes here, parental gametes. Gametes here, in the process that leads to formation of gamete is meiosis. Then we shall form T, then small t. Then here also we have capital T, small t. Very good. So let us now look at how there is going to be random fertilization. This is now random fertilization. We have capital T, capital T. Then there is this capital T, small t. Then we have capital T, small t. Then the organism here also we have small t, small t. This is fertilization, fertilization, which we should also have included it here. Fertilization should have included it there. Then semicolon. Fertilization. These are the genotype of the F2 generation. That is F2 generation genotype. F2 generation genotype. F2 generation genotype. Now we want to look at F2 generation phenotype. Phenotype. F2 generation phenotype. Here we have now this organism is different from this organism. This offspring, they are two different. This is different from this is different from this one is also different, and then this one. So these two are the same from the way you see the letters. This one is far different, this one is different from the rest also. So these organisms will appear tall. This one will also be tall, this one will also be tall, but this one here will be dwarf because it is lacking the allele controlling the tall condition. All the allele. So this organism is homozygous dwarf. This one is homozygous tall. This one is heterozygous tall. I repeat, this organism is homozygous tall. The two homozygous tall may be homozygous tall. These are heterozygous tall. Zygous tall. This is homozygous dwarf. Homozygous dwarf. So if you are to look at the ratio of tall to dwarf organisms, here we have F2 generation phenotype. We are going to have 3 tall to 1 dwarf. That is the ratio we have got. The ratio which Mendel got. So this is how this Mendel work can be represented by use of symbol. By use of symbol. So there are so many characteristics which they show monohybrid inheritance. So Mendel law show monohybrid inheritance, which I can put there as NB. Mendel first law is of monohybrid inheritance. Is of monohybrid inheritance. This thing of monohybrid, what does that mean? For me, this is characteristics of only dealing with one characteristic at a time. One characteristic at a time. Dealing with one characteristic at a time. One characteristic. A time. One characteristic at a time. Now, this is what This is what I feel, dealing with one characteristic at a time. What do I mean here? Here, the characteristics which Mendel deal with at a time here is the height of a plant. Height. Height has alternative form. Dwarf, tall. Those two are not characteristics. The characteristic is the height, or the trait there is height, but it has two alternative form. It has two alternative form. The alternative form is the dwarf and like you remember, dwarf and tall. You remember the tongue rolling. Tongue rolling also, I told you the tongue rolling, we also had got two characteristics in tongue rolling. Tongue roller and then non-tongue roller. So I would like you to look at this. So the characteristics which obey, the characteristics, the monohybrid inheritance in human, if we are to look at examples. The examples of monohybrid inheritance in human, we have got tongue rolling is one of them. So let me briefly give you example. Then you do these examples I have given you. You do the methods I've given you. The most important thing I want from you here, number one, is using this side here. You must write the wording at the left hand side. If you don't write, that means your work here, even if you do what, it will not work. So examples of monohybrid inheritance. Monohybrid inheritance. One is tongue rolling. Two, we have albinism. Achondroplastic dwarfism. And many others, etc. Now, I would like you as an assignment to state the differences between mitosis and meiosis. Mitosis and meiosis. That is it. I will come back. The next time I come back, I will want you to have found out that. And then we do the presentation, the answers, questions on this kind of thing. What we are left with are questions and only two other concepts, and we shall say genetics is complete. I would also want you to, maybe before I forget, briefly, I want you to state the reasons why genetics are studied. Importance of genetic studies. The importances of genetic studies. Those are, I think, enough, and I think we have come to the end of our lesson. I thank you very much once again to always be tuning to UBC as your public broadcaster, and with me, and you've been attentive to me several lessons, and I thank you, and several times. I wish you well, stay safe, nice time. Thank you very much.